Hello everyone, today we are going to learn about plant tissues. In the ontogeny of plants, groups of cells with the same origin, same function, and similar morphological structure are called tissues. There are many kinds of plant tissues. According to the difference of physiological function and morphological structure of plant tissues are divided into meristem tissues and mature tissues. Now, let's look specifically at two types of plant tissue, starting with meristem. Meristem is a group of cells in a plant that have the ability to divide continuously or periodically. It has the following characteristics. 1. Strong meristematic activity, typically located in the growing regions of the plant. 2. Cells are small, isodiametric, and tightly packed. 3. Thin cell walls, dense cytoplasm, and large nuclei. All other tissues of the plant are produced by meristem. According to their distribution, they are divided into apical meristem, lateral meristem, and intercalary meristem. They are responsible for continuous elongation of roots and stems, thickening of roots and stems, and rapid growth of certain parts over a short period. According to the source, they can be classified into protomeristem, primary meristem, and secondary meristem. The protomeristem consists of cells retained from the embryo, distributed in the apex of the root and the apex of the stem. Primary meristem is derived from the protomeristem and is located just behind it. Secondary meristem is formed by some mature tissues regaining the ability to divide. Next comes mature tissue. Mature tissues are most of the cells produced by meristem. After growth and differentiation, mature tissues gradually lose the ability to divide and form various tissues with specific morphological structures and physiological functions. According to different morphology, structure, and function, mature tissues can be classified into protective tissue, parenchyma tissue, mechanical tissue, conducting tissue, and secretory tissue. Let's start with protective tissue. Protective tissue covers the surface of the plant body and plays a protective role. It has the effect of preventing excessive transpiration of water and resisting external factors like wind, rain, diseases, and insects. It can be divided into epidermis and periderm according to different sources and morphological structures. The epidermis generally has only one layer of cells, and the outermost layer of cells in the leaves, flowers, fruits, and young roots and stems of plants are epidermis. It is classified as primary protective tissue. Periderm is a composite tissue with strong protective function. It is a composite tissue with strong protective functions. It belongs to secondary protective tissue. The second type is parenchyma tissue, named for its relatively thin cell walls. It is the basic component of the organs of the plant body, so it is also called the basic tissue. Parenchyma is divided into 5 types according to its function. Absorptive tissue mainly absorbs water and nutrients from the outside world, like the root hair cells on the surface of the root tip. Assimilatory tissue cells contain chloroplasts, which can carry out photosynthesis and are distributed in the green parts of plants. Storage tissue mainly stores nutrients, such as starch, protein, and so on. It is found in fruits, seeds, stems, and roots. Parenchyma has large intercellular spaces, often forming air cavities or channels, and serves a ventilation function. It is commonly found in aquatic and hydrophytic plants. Transfer cells function in short-distance transport of substances. They are commonly found in the minor veins of leaves and at stem nodes. The third type is mechanical tissue, which provides structural support. The main feature is partial or total thickening of the cell wall. According to cell morphology and the way of thickening, it can be divided into 2 types, parenchyma and sclerenchyma. Parenchyma cells are living cells, typically elongated and prismatic, with localized thickening often at the cell wall corners. Sclerenchyma cells are generally dead cells with small cell cavities and uniformly thickened cell walls. It can be divided into fiber and stone cells according to their morphology. Fibers are elongated, tapered at both ends, slightly spindle-shaped, and widely distributed in plants. Stone cells are short, wide, irregularly shaped, as shown, with extremely thickened, lignified cell walls, and are found in plant stems, leaves, fruits, and seeds, like the flesh of a pear. The fourth type is conducting tissue, consisting of tubular cells, specialized for long-distance transport of water and organic substances. This differs from the transfer cells mentioned earlier. According to the different substances it transports, it can be divided into 2 categories. One includes vessels and tracheids, which transport water and inorganic salts, and the other includes sieve tubes and sieve cells, which transport organic nutrients. Vessels are composed of many long, tubular, lignified dead cells connected end to end. Due to their larger lumen, vessels have a much greater transport capacity than tracheids. Based on their secondary wall thickening patterns, they are classified as annular, spiral, scalariform, reticulate, and pitted. Tracheids are the water-carrying tissues of most ferns and gymnosperms. Because of the different ways of secondary wall thickening, they can be categorized into the same 5 types as vessels. Sieve tubes are living, tubular cells, marked as Part 1 in the figure. Sieve tube elements have sieve pores at their ends. The end wall containing sieve pores is called the sieve plate, that is, the number 2 part in the figure. Sieve tubes are typically accompanied by one or more small cells called companion cells, marked as part 3 in the figure, which have a weaker transport capacity. The fifth secretory tissue refers to cells or cell groups in some plants that secrete substances like essential oils and resins, as well as latex, nectar, or mucilage. According to whether the secretion is excreted, it can be divided into exocrine structure and endocrine structure. Exocrine structures expel secretions from the body, as shown in Figure 1, including glandular hairs and hydrathodes. Endocrine structures and their secretions remain within the plant body. Figure 2 shows the secretory cavities for volatile oils in citrus peel and leaves, and Figure 3 depicts resin canals, such as those found in the stems and leaves of conifers. This figure is the transverse section of the plant stem. The outermost layer is the epidermis. Moving inward are parenchyma and sclerenchyma. On the left are bundles of conducting tissue, interspersed among other tissues. The larger celled areas represent the ground tissue. Therefore, the tissues in the plant body do not exist in isolation, but closely cooperate with each other to perform various functions, so that the plant body becomes an organic unified whole.